Hello everybody. Today we are going to be learning about a science experiment called the Visking Tubing experiment, which relates to diffusion and osmosis. So if you could first please do the usual, get your date and title down, have a look at the learning journey here. Have a look at the lesson objectives here, your keywords here, and your do now task. The video can be paused while you're doing that. Okay, moving on then. So let's just talk through the do now task. So a piece of potato placed in pure water is going to absorb more water than one placed in 20% salt solution. And that's because pure water has a higher concentration of water compared to the inside of a potato cell than our 20% salt solution. So water will move by osmosis from the high concentration to the low concentration, so down the concentration gradient. If you had a go at challenge, the words hypo and hypertonic refer to the solution. So our 100% water, our pure water, is more hypotonic because it has a very low, in fact, no salts dissolved in it. So it's more hypertonic compared to the solution inside the cell than our 20% salt solution is. You might also have said that the 20% salt solution is hypertonic compared to the pure water. Okay, moving on. So hopefully you remember that when we talk about osmosis and diffusion, we're talking about movement of particles across a semi-permeable membrane, i.e. the cell membrane. And it's got tiny little pores or holes in it that let certain substances through. So large substances like starch can't get through. Small substances like water, oxygen, glucose can get through. The Visking tubing experiment that we're going to talk about in a minute it sort of models what's happening with the cell membrane. Visking tubing is a material that's actually used in kidney dialysis, and like a cell membrane, it is partially permeable. So it lets water, glucose and oxygen through, but it doesn't let bigger molecules like starch through. Just to remind ourselves, oxygen, glucose and amino acids are all substances which enter cells because they're useful, but we talk about diffusion of those substances because they're not water. Substances that leave a cell by diffusion would be the waste products, so carbon dioxide and urea. So diffusion is really, really important because it lets the useful stuff into the cell, the waste product out of the cell. Osmosis is a form of diffusion. It is osmosis of water. So your first task is to have a look at these diagrams of cells and work out whether water will move from cell X to cell Y or from cell Y to cell X. Now in the first three pictures, A, B and C, the percentages you can see represent the percentage of sugar in the cell. In the last question, question D, you can see that the percentages refer to the percentage of water in the cell. So use the knowledge you've got about osmosis and what it means, and work out which direction water will be moving in each example. The slide can be paused while you do that. The video, sorry. Okay, so I'll go through the answers. So in question A, water moves from cell X to cell Y because water moves from an area of high concentration to an area of low concentration. Now our numbers represent the sugar concentration. So if we've got a low concentration of sugar, that's a relatively high concentration of water. So in A, the water moves from X to Y. In B, we go from Y to X, higher concentration of water here, lower concentration here. In C, we're talking about, again, this is still sugar concentration shown by the numbers. So water moves from X to Y, high concentration of water to low concentration of water. And X to Y again for D, because we've got high concentration of water here, lower concentration of water here. Okay, we're going to have a look at the Visking tubing experiment now. So this, as I said, the Visking tubing really kind of models or shows us what actually happens to substances moving across a cell membrane. So Visking tubing is dialysis tubing, as I said. It's semi-permeable, just like a cell membrane. And the experiment we do in school, very slightly different to this one. But in this one, they fill some Visking tubing with a sugar solution of different concentrations. And they place that in water and observe what happens in terms of water moving in or out through the Visking tubing and sugar moving in or out through the Visking tubing. They colour the water red so that you can see which way movement is happening. Get me out of the way. Oh, there we go. Okay, here we go. So sucrose is a type of sugar. So just to be clear, because they've gone through that really, really quickly, although the boiling tubes are labelled 0%, 20% and 100% sucrose solution, the boiling tubes actually contain distilled water that's been dyed red. The sucrose solution is inside the Visking tubing. Okay, so in our left-hand boiling tube, we've got Visking tubing containing water in a boiling tube of water. In the middle tube, we've got 20% sugar solution in the Visking tubing, and that's been placed in pure water. And then in the right-hand one, we've got 100% sugar solution in our Visking tubing, and that's been placed in water. Okay, so watch carefully what happens now. You're looking for movement of the red liquid. Do you see what's happening to the amount of water in the boiling tube? The red water. You don't need to write those down. Okay, so what they've done here is take out the Visking tubing and emptied the contents of the Visking tubing into measuring cylinders so that we can see what's happened. So with the 0% sucrose, it started off with 10 mils of water. We've still got around 10 mils in the Visking tubing. So nothing much has happened there. And we'd expect that because we've got water outside the tubing, water inside the tubing. There's no concentration gradient, so nothing changes. 20% sucrose, you can see that the water level has gone up from 10. So some water has moved by osmosis into the Visking tubing. In the last example, the volume of water has increased significantly. It's gone from 10 mils to millilitres to about 17 by the looks of it. So a significant amount of water has moved into the Visking tubing. And again, we would expect that because we've got a higher concentration of water outside of the Visking tubing and it moves by osmosis into our sucrose solution, which has got a very low concentration of water at the start. So a hypertonic solution has moved by osmosis into the hypertonic solution. Okay, I'm going to stop that there. Okay, now we're now going to look at an example of another practical, which is very similar to that. And you're going to think through and make predictions. Okay, I've just remembered that this needs moving, otherwise it's not very clear. Okay, I'll clarify that in a moment. Okay, so if we were doing this in school, this is what we would do. We would collect a piece of Visking tubing. That's this stuff. And we would tie a knot in one end. Now into that Visking tubing, we would put 6 centimetres cubed of glucose solution, which is just another type of sugar solution, and 6 centimetres cubed of starch solution. Okay, so what we end up with is a mixture of sugar and starch solution in here. These labels are not very good on this slide, folks. They should be pointing into here. This is the sugar and starch. Okay, we then clip or tie the top end of the tubing so that our sugar and starch solution can't just leak out the ends. We then place our Visking tubing, our sausage, into a boiling tube filled with pure water. So this is pure water, 100% water, or 0% glucose solution or starch solution. In here, we've got glucose and starch dissolved in some water. As soon as we've done that, we can carry out a test on the water to make sure that it's pure. Okay, so these are food tests. I'm not sure if you will have done these in year 7, probably not, as we had lockdown. So we can carry out a test called the Benedict solution test on our solution, and that tests for glucose. So if glucose is present, we would see a colour change from blue to orange. Okay, So at first, when we test this, we would expect not to see a colour change because we know that this is pure water. We can also do something called the Starch Test, where we use a reagent called iodine. Now, when you add iodine to a solution containing starch, you see a colour change from brown to blue-black. Now, again, at the start, we wouldn't expect to see a colour change because we know that at the start, this is pure water. What I want you to think about is what might happen, what might we see if we repeated these tests on the water that's outside the Visking tube after 20 minutes. Now remember, the Visking tube is semi-permeable, like a cell membrane. So it allows small molecules like glucose and water to move through it. It doesn't allow larger molecules like starch to move through it. So after 20 minutes, if we were to test this water with Benedict's solution and iodine, what would we see? And can you explain why we would see that in terms of osmosis and diffusion? So osmosis, the movement of water molecules, and diffusion, the movement of other molecules from a high to low concentration. So this slide will stay on the board. Your task is this bit here, completing a storyboard analysis of what happens. So can you show, using diagrams, what's happening in terms of movements or not movement of molecules? I want you to think about the water, the glucose and the starch and what we would find when we tested the water at the end. So you've got probably 10 to 15 minutes to have a go at doing that. The teacher can pause this slide while that's happening, and then afterwards I will go through what you should have written. Okay. Okay, so hopefully you've given that a good go. So these are the sorts of things I hope you will have come up with. There will have been no movement of starch molecules because the Visking tubing acts as a semi-permeable membrane like a cell membrane. It doesn't allow starch molecules to move through. So if we had have tested the water after 20 minutes, we would still get a negative result for the starch test. The iodine would not have changed from brown to blue-black. We would have seen no change at all. However, we would expect to have seen some movement of glucose molecules from this area of high concentration of glucose. They would diffuse outwards into the water by they would diffuse by diffusion, obviously. They would move by diffusion from the area of high concentration of sugar inside the Visking tube into the water where there was a lower concentration of sugar at the start. So if we then carried out the Benedict solution test for glucose on our water after 20 minutes, we would expect to see the presence of sugar in that water. You might also have shown that water will have moved by osmosis into the Visking tubing because we've got a higher concentration of water at the start here than here, and water moves by osmosis towards the lower concentration of water. Now, if you were really smart, you might have managed to use keywords hypertonic and hypotonic. So for glucose, we start with a hypertonic solution of glucose inside our Visking tubing. So diffusion takes place from hypertonic to hypotonic. Osmosis takes place from hypotonic to hypertonic. If you manage to get those keywords into your descriptions, then really, really well done. How is that relevant in living things? Well, we know that water will move from a hypotonic solution into a cell, such as a plant cell. And if a plant cell has got lots of water in it, we call it turgid. Okay, so water will enter the cell, causing it to swell up, and then we say it's turgid. If we have an isotonic solution outside a cell, then there'll be no change, no overall osmosis of water. If the cell is in a hypertonic solution, in other words, one with lots of salt or lots of sugar, then water will move out of the plant cell. Now, this doesn't actually make the cell shrink because the cell is supported by a cell wall, but the cell membrane and the vacuole shrink away from the cell wall inwards, and that's called plasmolysis. So we say the cell is plasmolysed. So that's a really good keyword to jot down and remember. So this could, if you want to pause this now, you could just make some notes on what happens in plant cells. And now let's think about what would happen if we placed blood cells in different solutions. So an example of this might be somebody being given a drip in hospital where a solution is being injected into the veins. So if we were to give somebody an isotonic solution, a solution that's exactly the same concentration as the red blood cells, then we would expect no change, no movement of water in or out of the blood cells because there's no concentration gradient. If we were to put a hypertonic solution into the veins, so a high salt or sugar, a high concentration of salt or sugar into the blood, then the cells would actually lose water because the cells would have a higher concentration of water than the hypertonic solution. The cells would lose water and shrink. That would be really damaging for them. Equally, if we have a hypotonic solution, then a higher concentration of water outside the cell than inside the cell could cause the water to move into the cell to the extent that it bursts. So getting exactly the right concentration of solutions when we give people things like drips is really, really important. Okay, your next task is to have a go at explaining that using this slide to help you and using this sentence starter at the bottom to help you, So for each situation that a red blood cell could find itself in, write a sentence explaining what's happening in relation to osmosis. So for example, when a red blood cell is placed in a hypotonic solution, it bursts because water is entering the cell, moving from a high to a low concentration. Okay, so see if you can write a sentence for each of those three scenarios, and that is your last task for today, I think. Yes, it is. So that can be paused while you do that, and then I will go through the answers. Okay, so you should have said that a red blood cell placed in a hypotonic solution will burst because water will move into the cell, or water will enter the cell, moving from a high water concentration to a low concentration. When a red blood cell is placed in an isotonic solution, there is no change because the concentrations are equal in and out of the cell. And when a red blood cell is placed in a hypertonic solution, water leaves the cell in order to move from an area of high concentration to an area of low concentration, and therefore the cell shrinks. I hope you found that okay. If there was anything you struggled with, perhaps take a moment to jot down anything you found difficult or any questions you have for when we're back in school together. Okay, thank you.